Hello and happy Earth Day. Today we're going to be talking about environmental pollutants, really, and their impact on development, human development, on Earth Day 2020. So this is chapter 23, Development in Health and Disease. We're going to focus on teratogens and in the environment. Got some slows to get you started. I probably need to add more to those. We are focusing on part 2 of this 5-part series. This is teratogenesis, environmental assaults on animal development. So teratogens is Greek for monster formers, and you can look in the glossary of your book for a more thorough definition. And you can also look in your notes of your PowerPoint slide because your previous one, part 1, I have the definition there. And one more time, I have the definition here. So teratogens are exogenous agents. That means they're outside of your body that cause disruptions in development resulting in what we call teratogenesis. So that's development in the presence of teratogens. And you get the formation of what are called congenital defects. Congenital defects are ones that you're born with. Teratology is the study of birth defects and how environmental agents disrupt normal development. So teratogens were discovered in 1962 to 1964, and this lady here, Rachel Carson, was among the folks who called attention to the impact of the pollutants on our environment. And she wrote a book called Silent Spring, and naturally as a woman she was chastised and ignored, but she was absolutely right. And she focused on DDT, which is a chemical that was used to reduce the spread of malaria, for example, by killing the host. And the host is mosquitoes. And she found that it was impacting birds and making the shells of the eggs very soft. And so when the mothers would nest on top of their eggs, they would actually crush their babies. It looks like you can see the second D and the T, but that's DDT. When you pull up your PowerPoint, you'll be able to read that. We've also mentioned in the past the effects of thalidomide, which was a drug used on pregnant women to reduce nausea. And it had severe impacts on these children that were developing in utero. They were fetuses in development. You can see that they've got what is called phocomelia, that is shortened limbs. And these kids have shortened arms in particular. Another teratogen besides DDT and thalidomide are infectious agents such as rubella measles. So this child here was born with congenital blindness and deafness because his mom had rubella measles. This is a reason that we have vaccines against rubella measles or German measles and other measles and so forth. Because before vaccines, we had so many people that would suffer these types of infections. I mean, pandemic was a normal sort of condition in a sense. At least lots of, I know my mother and my aunt, my grandma, they all suffered from mumps, measles, those kind of things, scarlet fever. There were a whole host of people that suffered from polio. But then we had science and vaccines, and we eradicated a lot of these infectious agents and a lot of these things that really impact human health and development. But now we've got anti-vaxxers who think that the it's all a conspiracy, or that, I don't know, that you should just rely on innate immunity. Well, we're seeing right now pretty drastically what the world looks like when we don't have vaccines against something such as COVID-19, and it's not a pretty picture. So teratogens are things in the environment that specifically impact organisms that are undergoing development because they're very sensitive. They've got rapid cell division, as we've talked about several times during the class period. Those early stages of cleavage, we've got lots of DNA replication, we've got lots of cell division, and so embryos, fetuses are much more susceptible to chemicals in our environment and to infectious agents in our environment. From your chapter 23 in the 12th edition of the book, it's chapter 24 in the 11th edition of the book, there is a chart, a figure, weeks of gestation and the sensitivity of embryonic organs to teratogens. So I want you to study this chart. In the cleavage stages of embryogenesis, embryogenesis is the first 8 weeks of pregnancy. The first 2 weeks of pregnancy is where we've got this rapid cleavage stages, implantation, and gastrulation occurring in humans. If you're exposed to teratogens during this period of time, your malformation is going to result in death. However, if you're a little further along during embryogenesis in weeks 3 through 5, you are going to have major impacts on whatever organs are developing during this period of time, and these are shown in pink. These are major congenital anomalies will result if you're exposed to a teratogen during weeks 3 to 5 of embryogenesis. And you can see here in these little black dots that there are common sites of teratogen action, including the heart, central nervous system, eye, there's heart again, and limbs. Okay. Now, continuing in embryogenesis, you can still continue to have major congenital anomalies result when you're exposed to teratogens during this 6 to 8 week period. And you can see again by the black dots that there are common sites of teratogen action, including now the teeth and ears, which did not exist during these first 3 weeks, the palate, okay, forming your mouth. And your brain is still in the picture, CNS, right? CNS, brain, always affected during embryogenesis if you're exposed to a teratogen. And now in the last 2 weeks of embryogenesis, weeks 7 and 8 of human development, external genitalia are also impacted. So if you're exposing your embryo developing in your uterus to external genitalia, to teratogens during weeks 7 and 8, you're going to affect your child's external genitalia development, for example. All right. Now, starting at week 9 of human development, we no longer call the developing organism an embryo. Now we call it a fetus. At week 9, it is a fetus. And you can see that the central nervous system is still sensitive to teratogens. It has been sensitive the entire time so far. Now, at the end of week 16, that is the end of the first trimester. So everything we've talked about up to this point is the first trimester. And then after that, you've got fetal development. If shown in yellow, you have exposure to teratogens, you have functional defects and minor anomalies. So you can cause birth defects still by being exposed to teratogens at this period of development, but they're not as severe. So here's a quote from Rachel Carson, which I liked and your book likes, and it's in your book. It is all of a piece. That means she's saying it's all one whole thing. Thalidomide and pesticides. They represent our willingness to rush ahead and use something without knowing what the results will be. So here's a chart, a table from your chapter 23. This is 23.1. some agents thought to cause disruption in human fetal development. So from this table, I'm going to point out, we're talking about these that I'm highlighting with red arrows, or your video talks about them, your assigned video. So you should know those. And then the ones in blue, we're not talking about in depth, but I am pointing them out because I think you should know them. And one of them is retinoic acid. Your book does have a section on that, but I'm not covering it. One of the things here is Accutane, and I know that's used a lot to treat acne, and so you might want to lay off your acne treatments if you're thinking of having a child. Another one is cortisone, and of course lead, and then we've got X-rays, which you probably knew about, and fever, hyperthermia. This is why it's important, again, for women who are expecting to be vaccinated or to avoid getting sick, like, what we're doing now, staying at home to avoid COVID-19 infection that could cause fever. Because fever by itself can impair human fetal development. It can disrupt it. And then I have these in black, which I just wanted to point out that, we take antibiotics almost like they're candy to treat infections. But look, penicillin, tetracycline, and streptomycin, all three of those are antibiotics, and they disrupt human fetal development. So keep that in mind. I forgot to point out diabetes down here. So metabolic conditions in the mom can affect the fetus that's developing within her. And for those of you who took genetics, you'll see phenylketonuria is in here too. Now I'm going to move on to Zika infection during fetal development. This causes what we call microencephaly, and we're going to look at the mechanism that has been revealed. So there was a big scare in 2016 when the World Health Organization announced Zika, and it was in more than one country. So it didn't go all the way around the world to classify it as a pandemic, but it was infecting a number of countries, including those that we plan to hold the Olympics and the Summer Olympics down in Brazil. And so there was a big controversy. Should we still have the Olympics? Well, they did. And, that's what could happen, is that you could be infected by Zika virus. So this was in 2016, just 4 years ago. In Geneva is the headquarters of the World Health Organization. And of course, our current president wants to withhold funding to that organization because he wants to blame them for not giving the United States a big enough heads up when he actually ignored their warnings and their offer for help to give us the formula for making test kits. So that's the situation we're in now. But remember, that was just 4 years ago, 2016. CDC whirled on Zika virus as a teratogen. So CDC is our, Centers for Disease Control, run out of Atlanta, Georgia. And they gave a big heads up to pregnant women, saying that if you had infection during your pregnancy, you could affect the development of your child, and the child could come out with a head that's too small, which means that there's a brain that's too small. So, and in particular, wanted women to avoid or be preventing mosquito bites, because that is how you could get Zika. And so here's the mosquito shown here. Here's a pregnant woman here shown here. And then you can see that there are other images of doctors and scientific methods and childbirth. And using mosquito repellent, for example. Now, you could get Zika and not really have any symptoms. And, that's the same thing that's happening with COVID-19. And COVID-19 is also imperiling the possibility of the 2020 Summer Olympics. So I see some similarities. But what is microcephaly? So microcephaly is when the baby has a head that's smaller than usual. So these images, this little educational tool, is brought to you by your Centers for Disease Control, which is funded by your tax dollars if your politicians allow that funding to go to them. There's been cutbacks. So here's a baby with a typical size head shown here on the left. Here is a baby with a microcephaly, and you can see that in the dashed lines, they're showing you what the head size should be. And then here's a baby on the far right with severe microcephaly, and you can see how much smaller that head is compared to what normal size is. Now, not only is it not nearly as attractive, but it's also functionally bad news. So here's a lady getting possible Zika. So things that could happen with a Zika infection but don't always happen are fever, rash, joint pain, and red eyes. Zika virus triggers increased expression of a microRNA that decreases expression of a nerve growth factor. And this is now in your textbook. So this was just something we discovered 4 years ago, and you've got it in your textbook now, which I think is remarkable. Usually textbooks are like 20 years behind science. Figure 23.3 looks at the possible mechanism for Zika virus-induced cell death in those babies' brains. So what we're looking at here is a figure that's going to show us that Zika infection upregulates the microRNA known as MIR-9. So why would it upregulate that? Well, for those of you who took genetics, remember, we've got RNA interference. That's the cellular immune system. So a virus comes in there, and that's going to activate your microRNAs or your immune system, so forth. Usually that's siRNAs, but in this case, it looks like we're upregulating a microRNA. So shown here on the y-axis is the relative amount of that microRNA called MIR-9, and you can see in control developing brains, you have very, like a relative amount of 1 on this scale. In an infected baby's brain, you can see that it is significantly increased. It's 4 times greater, 3 to 4 times greater in the infected individual, this expression of this microRNA. And this is a significant difference as indicated by the p-value here of 0.001. Anything that's 0.05 or less is considered significant. Moving along, how does this impact fetal brain development? Well, because we see that increase in that microRNA, we also see a decrease in a factor important for neural growth, and that is called GDNF. So this is what the MIR-9, this microRNA, targets. It's going to regulate the expression of this nerve growth factor called GDNF. And see on this graph here, we've got relative amount of GDNF on the y-axis. We've got a relative amount in your control. So normal expression is relative level 1. If you are an infected brain, you have this going on. You've got your expression of this growth factor severely decreased. Almost down, well, it's decreased by a factor of 2, it looks like. It's almost down from 1 down to about 0.5. It's getting there. All right, so that's what's happening. We're decreasing the ability of the fetal brain to grow. And as a result, we decrease the number of cortical neurons that are in that baby's brain. So normally shown over here in this left panel, the control, we've got cells that are labeled blue. Those indicate that we've got a lot of cells in these locations, because each cell has a nucleus that's labeled with blue. In this green section, you can see the number of cortical neurons that you should have in green. And then you can see in the infected individual the number of cortical neurons that you have. And you can see that they're decreased by at least a factor of 2. So half the amount of cortical neurons in a Zika infected brain compared to a control brain. And that is why you get smaller brains and then you get smaller heads. So I found this from a Nature article that I thought was really cool, since we've talked about organoids in class. Organoids were used to study Zika because we don't want to just take babies and infect them and see what happens. So we're creating organoids of the brains to study Zika's mode of infection, and it seems to affect a particular type of cell called the neural progenitor cell. So you guys remember that progenitor cells are somewhere in between being a stem cell and being a differentiated cell. So they looked at the infection found in the fetal cortex. And on the left, we've got Zika virus infecting a cell. So this would be a human neural progenitor cell. And the virus is expressing as a single-stranded RNA genome. Then it's going to replicate itself. So we're going to have RNA replication. And then Dicer is going to recognize this double-stranded RNA and cut it up and do this jazz and try to fix it. But it's in these neural progenitor cells. And so what's happening in these cells as they're battling this virus is that they're focused on battling the virus instead of doing their job, which is to replicate and create cortical brain cells, neuron cells. So what's happening here in this brain organoid is that the Zika virus is going to cause organoid shrinking and structure disruption. However, they found that there is some sort of broad spectrum drug called enoxaparin that can restore normal development in the presence of the Zika infection. So there you go. There's a case where you're using some sort of drug to actually help development. Shocking, I know, but it's possible. So here's where in the world, according to the 2019 map, Zika is located. And you can see it's in the United States, shown in purple on this map. So this is a country or territory that has ever reported Zika cases, past or current. And we did have some infections. People brought those home from Brazil. You know, I'm not a big world traveler, so it's really the wealthy. I blame the wealthy. And then maybe people like Olympics folks and students who study abroad, who have money or get money, raise money to travel all over the place, that then spread the disease throughout the world. And then the rest of us can possibly get it. Isn't that fun? So here's the area of current Zika outbreak in 2019, which is in India. So it's not gone. It's just not as bad. We get immunity and it's not as bad. And we find ways to treat it and it's not as bad. But it's still out there. So COVID-19 in the fetal brain. Well, we don't really know. It's unknown, but I'm thinking it needs to be investigated. CNS involvement has been noted as a symptom of COVID-19. There are reports that include loss of taste and smell, strokes, cognitive decline, and dementia. Now, this is not in everybody. Remember, there's multiple strains of the COVID-19 virus out there, and everybody's got a different genotype. So those things impact how you experience the infection. So this is just evidence for what I just said. This is from a journal in 2020, March 2020. Coronavirus not only affect the respiratory system, but also have deleterious effects on the central nervous system. Most neurological diseases could be caused by a coronavirus invasion. Coronavirus causes nerve damage via diverse pathways. Okay, so nerve involvement. And this is from the journal Neuralgia. That was from Brain Behavior and Immunology that I just quoted. Neuralgia, another journal, I just pulled from their abstract this little bit that says, conclusion, although neurological symptoms are not frequent in coronavirus epidemics, the high number of patients with SARS-CoV-2 infection may explain the presence of the virus in the CNS and increase the likelihood of early or delayed onset neurological symptoms. Follow-up of patients affected by this, what's really COVID-19 epidemic, should include careful assessment of the CNS, the central nervous system. So somebody should investigate whether it affects developing embryos, fetuses. It's unknown at this time. But since it can cause most neurological diseases, you just have to check it out. So I already sent you this assignment. You should watch Scientist Speaks 23.1 on your textbook website, Environmental Toxins and the Embryo. And one more thing. See the slide notes for questions to answer while viewing the approximately 25-minute video. Use your notes to study for exam number 4. So food for thought, or I. Talk a little bit about sonic hedgehog signal disruption. So hedgehog is one of those 4 paracrine factors that we talked about when we talked about morphogens and how important morphogens were for signaling. So this is what happens when you disrupt sonic hedgehog in a developing lamb. So the mom ate this wonderful plant over here called Veratrum californicum, and it contains a couple of special ingredients that are teratogens, and those are gervine and cyclopamine. And of course, what results is that the developing fetus in the mom that ate this plant ends up with one eye. This is the head of a cyclopic lamb born of a ewe who had eaten some yummy Veratrum californicum early in pregnancy. And it disrupted the sonic hedgehog pathway and resulted in this birth defect. So just a reminder, cholesterol is part of what slows down the diffusion of this morphogen and limits the number of cells it signals. Cholesterol is kind of like a parachute for this paracrine factor, so that it affects only the cells it's supposed to by turning on transcription, but not cells that are further away. Unfortunately, when you eat gervine-containing plants, this cholesterol attachment is not put on, and so sonic hedgehog diffuses through the embryo faster and affects more cells and turns on transcription at the wrong time. So it's turning on transcription at the wrong time, and that's what gervine did for that lamb. So moving on to oil and water really don't mix. Deepwater Horizon 2010 oil spill, which happened in our neighborhood, the Gulf of Mexico, and affected the coastlines of Louisiana, Mississippi, Alabama, and Florida, and Texas to some extent. This is what the water-soluble components of that crude oil did to zebrafish. So normal development is shown on the left, abnormal development is shown on the right. You can see that the head and thorax are definitely not formed correctly. It's affecting neural crest cell migration, and so we've got malformed cranium. So this happened in 2010, right before Earth Day in that year, 10 years ago, April 20th. 200 million gallons of British petroleum oil was spilled from one of their rigs that we have out there in the water. This is why it's not the best idea to pump oil out of the ocean, but hey, that's what we do. 87 days of the spill, it just was pumping it out, and it was quite horrible. I remember it well. And 16,000 total miles of coastline affected. Well, if you don't care about anything else, it affected your economy there because people didn't want to go to the beach. People also didn't want to eat whatever was coming out of that water. So there you go. Food for thought of a person of business mentality. Alcohol as a teratogen. Y'all knew this, but here it is. Fetal alcohol syndrome, FAS. And there's also fetal alcohol spectrum disorder, or FASD. So normally you've got this sort of development, and yes, I'm sorry it's Caucasian, but this is what they had on Adam, the website where you get these pictures. So you've normally got an inner canthus that looks like this, and you've got, everybody has this, a philtrum, which is that little ridge that goes between your nose and your lips. And then you've got a nice nasal bridge and so forth and so on. When you have fetal alcohol syndrome, you've got different phenotype. You have the absence of that philtrum, for example. You have flat nasal bridge instead of one that's open. You have epicanthal folds instead of canthal folds. So those are some of the hallmarks, and you can see that these faces look a little bit different. So comparing fetal alcohol syndrome to fetal alcohol spectrum disorder, we've got a small head resulting from fetal alcohol syndrome. Again, much like Zika, alcohol affects the brain development. You've got less brain mass, you've got fewer brain cells, so smaller brain, smaller head. Indistinct philtrum, narrow vermilion border on the upper lip, low nose bridge, lack of brain development, which we're going to look at in a little bit more detail, and most prevalent type of mental retardation that is known. 1 in 650 children, that's a pretty high rate, have this, are affected by fetal alcohol syndrome due to drinking alcohol during pregnancy. The mean IQ of these kids, and that's the mean, that's the average. So there are some that are higher and there are some that are lower. Right? This is the average or mean IQ is 68, and I'll talk to you in the next slide about what that means. There, for example, a 16.5-year-old adolescent would have the vocabulary of a 6.5-year-old child. So that's how severely this IQ can affect you, and this is, of course, how this teratogen known as alcohol can affect a developing fetus. So phenotype, adults often cannot handle money and difficulty learning from past experiences, and also difficulty just talking in some, it depends on how severe it is. Fetal alcohol spectrum disorder, term coined to encompass all alcohol-induced malformations and deficiencies that occur. Behavior abnormalities, not always physical. They can be IQ associated, but substantial neuronal deficiencies and executive brain function. This is their phenotype, including reduced memory, reduced ability to plan, decreased information retention. So it can affect your ability to learn. Estimate, for every 1 case of fetal alcohol syndrome, there are 3 times more cases of fetal alcohol, spectrum disorder. I switched my S and D there. So IQ, average is about 68, as I said, for those who develop